So, well, first of all, thank you for inviting me, Michal and Helena, to present to this group. And I was sort of encouraged to think in sort of a broad way about evolution and adaptation in toll-like receptors. And in some ways, I sort of slightly regretted it when I started putting the talk together. So what I'm going to try and do is to give a broad overview of the types of pressures that exist and the types of signals that we can detect. But in doing so, I will give you various examples of different aspects of evolution of toll-like receptors. Now, what's clear is that I couldn't cover all of the ground, so I apologize ahead of time for any topics that you might be really interested in that I actually ended up omitting from the talk. So really, by way of background, I come at this problem very much as a functional immunologist that's interested in comparative biology. I'm interested in how different organisms actually use largely similar systems to combat wide ranges of pathogens, often in very different ways. So let's move on. One of the key things to this, and I think it's key to that sort of almost to the whole meeting, is how do we think about the immune system? So it's clear that immune systems and pathogens are in constant conflict, so we're going to be able to see adaptation and selection. But actually, intellectually, we tend to compartmentalize the immune system, and every meeting compartmentalizes the immune system. And in its broadest view, it tends to compartmentalize it according to whether it's considered part of an innate immune system or an adaptive immune system. Now, I have a little bit of an issue with this because actually, most of the genes that we're talking about are often considered really as part of both parts of the immune system. You know, many of the cytokines are involved in both innate and adaptive, and it's a highly integrated system. You don't get adaptive responses without innate immune responses, and often the adaptive immune responses operate by virtue of activating so-called innate immune responses. So what I wanted to do was in thinking about the sort of rules behind adaptation and selection, I was sort of trying to turn it a little bit on its head and think about the immune system as a bunch of receptors, a bunch of molecules, some of which are outward facing, i.e. they face the pathogens coming in, and others are more inward facing. And the outward facing molecules directly interact with pathogen products, and they might include the MHC, the pattern recognition receptors, some effector molecules like defensins. And often there we can see signatures of selection in the coding sequence, much more commonly than with other molecules. That isn't to say that selection doesn't act on some of the inward-facing molecules and genes. And that the problem is that most of the inward-facing genes are really constrained because they primarily interact with other self molecules, such as cytokines with their receptors. And that means their coding sequence is really constrained. And when we think about selection, or the selection that's effective in controlling pathogens, it's actually more often that we see changes in expression rather than coding sequence changes. So we're interested in pattern recognition, and we decided to pick up toll-like receptors many years ago. And I'm just going to try and take you through a little bit of a story in the next few minutes with the toll-like receptors. So what are they? Very basic introduction. I apologize for that for those that are already experts. They're an ancient receptor family present in as immune genes in vertebrates and in invertebrates. They're part of this sort of immune sensing army called the pattern recognition receptors, first proposed by Charlie Janeway back in 1988. And the toll-like receptors are pattern recognition receptors that survey the space that's sort of close to cells. There are other pattern recognition receptors that are found in the tissue fluids or in the cytoplasm. There are many more also on the cell surface. But the toll-like receptors specialize in surveying the space close to cells. They have a very conserved structure, a sort of typical question mark almost sort of structure with an extracellular domain that's involved in recognizing the pathogen bits, a transmembrane domain, and a signaling domain called a tear domain. Most vertebrates have between 10 and 12 toll-like receptors, and there's really quite broad conservation of specificity to a bunch of molecules that Charlie Janeway called pathogen-associated molecular patterns. So these Pamps that toll-like receptors see tend to be close or associated with the cell surface. What do they see? What are the agonists of toll-like receptors? Well, they see a range of agonists, and this is just a small selection. So, for example, they have specificity. TLR2 can be paired as a heterodimer either with TLR1 or TLR6 and see slightly different modifications on lipopeptides. TLR5 recognizes flagellin, the flagella subunit by bacteria. TLR4 recognizes a molecule on the surface of bacteria, lipopolysaccharide. Those TLRs that are found in endosomes are really the nucleic acid recognizing TLRs primarily, and they include TLR3, which recognizes double-stranded RNA, TLR7, single-stranded RNA, and TLR9, unmethylated CpG motifs in DNA. So they have a range of different specificities to different types of molecule that pathogens find it difficult, but not impossible, to check. So if we were to consider vertebrates generally, and I've just picked a simple comparison of chickens versus humans. Both chickens and humans have 10 TLRs. They have a clear set of directly orthologous TLRs, including TLRs 3, 4, 5, and 7, direct orthologs in both groups. They have TLRs that represent conserved family members. So both chickens and humans have TLR1 and 2 families. In humans, the TLR1 family is represented by 3 genes. In chickens, it is just 2, whereas in humans, the TLR2 family is represented by a single gene, and in chickens, and all birds represented by 2 TLR2 genes. What's clear that in chickens, what we don't find is TLR8 and TLR9. And TLR8 recognizes very, very similar agonists to TLR7, so maybe birds are getting away with just using a single-stranded RNA recognizer. But TLR9 was a bit of a problem because TLR9 in mammals recognizes unmethylated CpG motifs. And before we even started looking for TLRs, we already knew that in birds, unmethylated CpG was a really strong stimulator of immune cells. That problem was resolved. We looked very, very hard for TLR9 and couldn't find it, but that problem was resolved when 2 TLRs that are not found in mammals were identified. And the guilty party in terms of unmethylated CpG recognition is TLR21. There is, of course, this other TLR, TLR15, which is generally present in most reptiles and birds, and that is something I'm going to come to in the next slide. So what happens when you find completely new TLRs? Well, of course, it's key to do functional work to try and identify what they see. So with TLR15, the work I'm going to present on this slide, it's found in most reptiles and birds, but not in mammals or amphibians. It has been found recently in a shark. If you look at the TLR15 sequence and you separate it according to the signaling domain, the tear domain, then TLR15 really looks like one of the TLR1-2 family members. But if you look at the extracellular domain, the domain that's responsible for recognizing agonists for activation, then TLR15 is really very distinct from those TLR1s and 2s. So what we know is that the likely signaling profile was 1-2-like, but the agonist, we had no idea. So what did we do? We cloned TLR15 some years ago, and then we threw the kitchen sink at it, the microbial kitchen sink. We picked a wider range of agonists that we know stimulate toll-like receptors. We lysed every pathogen we could lay our hands on, and we kept coming up with completely negative results. Nothing was stimulating our TLR15. That was until the PhD student at the time, Amy Boyd, decided that she would try, I think the first one that she tried was brewer's yeast. So she tried yeast and fungal lysates, and found that this stimulated TLR15 really very strongly. Now, what we know is that this this ability to activate TLR15 is actually inhibited by heating the lysate or by treating it with the enzyme inhibitor PMSF. So we know it's an enzymatic activity within yeast and fungal lysates that's responsible for activating TLR15. At the same sort of time, other groups were also involved in identifying these sorts of agonists as being activators of TLR15. So one of the things that I hear about, pardon me, a lot of the time when I go to meetings is people talk about TLRs as if there are some TLRs that are antibacterial and other TLRs that are antiviral. I really want to very gently sort of say, perhaps we ought to change our language there, because TLRs recognize agonists, not pathogen types. So these TLRs some of them might seem to be focused on particular pathogen groups, but be really careful. So for example, and I put some examples down at the bottom of the slide, toll-like receptor 2, often referred to as a bacterial recognizing TLR, and it recognizes bacterial lipopeptides, but it's also involved in seeing yeast cell walls in mammals, and also protozoan GPI anchors. Well, they're not bacteria. TLR4, similarly, well known for recognizing lipopolysaccharide, or LPS, but also responsible for recognizing some viral agonists, such as RSV glycoproteins. This problem gets even bigger when we start thinking about the other major group of patterns that pattern recognition receptors recognize. And they're known as the damage-associated molecular patterns, or DAMPs. And many TLRs don't just recognize Pamps, they also recognize DAMPs. And it's like having a set of receptors that are capable of recognizing a problem is present in a wide variety of ways. And what is a DAMP? It's essentially recognition of inappropriate self. So maybe modified self or heat shock protein or whatever. And all of these TLRs are capable of recognizing DAMPs. The other point I wanted to make early on is that we all think about TLRs as being absolutely critical, and they are. If you don't have a TLR or TLR function, you can die of infections, but they're also incredibly dangerous. And I'm sure that many of you here are perfectly aware of a condition known as endotoxic shock. Well, endotoxic shock is lipopolysaccharide. Well, we've already talked about lipopolysaccharide. It's recognized by TLR4. And endotoxic shock is essentially too much endotoxin, too much LPS stimulating TLR4, and that can lead to really severe disease. So there are two, I picked the example I picked here are actually two mouse strains, two inbred mouse strains, the C3H on C3H backgrounds, the HEN strain and the HEJ strain. And the HEJ strain is quite famous because it has a point mutation in the signaling domain of TLR4, and it doesn't work. So if you intravenously inject LPS into these animals, the intact individuals have a really severe outcome. They get very sick and essentially die. Whereas the C3H HEJ mouse just runs around as if nothing's happened because it doesn't experience the endotoxic shock. However, if you give these same animals an infection with salmonella, the intact individuals respond quite strongly. They might get a bit sick, but they survive salmonella infections, whereas the C3H HEJs succumb to infection because they can't adequately activate the immune response. So this means that we need the TLRs to survive the infection, but overstimulation can kill. And under some circumstances, actually TLR gene loss or reduced signaling can actually be viewed as adaptive avoidance of pathology. And Stephen Fidderman later on in this session is going to present quite an interesting story with toll-like receptor 15. So TLRs, what sort of variation can promote survival? We have the obvious non-synonymous coding sequence variation. Most of it tends to be focused in this leucine-rich repeat domain. This is where the pathogen bits are being interacted with. Some is also found in the signaling domain, the tier domain. And that can have really quite dramatic effects to the way the TLR responds. You can get non-coding sequence variation, for example, in promoters, and that might affect expression levels or cellular distribution, but that's often not looked at. But the other thing that we have to remember is that changes in other things that genes can affect the way that TLRs work. So for a bunch of TLRs, there are actually co-receptors. So particularly with TLR4, which when interacting with lipopolysaccharide, it actually involves a complex of molecules, including MD2, CD14, and lipopolysaccharide binding proteins to optimally stimulate TLR4. So polymorphisms in any of those can actually also have effects on TLR4 biology. Downstream signaling pathways can be different between different species or between different individuals. And then, of course, we've got the target genes. Their promoters can actually be different, and that could lead to a different outcome from the stimulation of the same TLR. And as I've mentioned before, deletion can be beneficial. These are pretty dangerous receptors. Pardon me. So I now just want to move on to a couple of little case studies. And in this case, it's going to be with penguins. And you might ask, why penguins? Aren't they pretty tough to get samples? Well, they are. They live in pretty inhospitable places, a lot of them. So I've got to first express huge thanks to our resident penguinologist, Tom Hart, and other penguinologists. What a cool term for your job. So these guys that provide us with samples to try and answer some of these questions. But really, why penguins is that they occupy really diverse niches in terms of host pathogen interaction, in terms of the environment, the physical environment, but also in terms of the biotic environment. Some penguins are really quite isolated from other individuals and only get penguin pathogens, whereas other penguins are basically crapped on by everyone and get pathogens from all sorts of birds. And these environmental gradients that we're familiar with don't just affect the birds, they affect the survival of pathogens too. Some pathogens are more or less able to survive the harsh conditions than others. So let's think about penguin toll-like receptors. The first question is there lots of variation in penguin toll-like receptors? And the answer is yes, there's lots of variation. And a whole bunch of this variation includes non-synonymous polymorphisms. Now, most of the non-synonymous polymorphisms are in the extracellular domain of the toll-like receptors, with relatively few in the signaling domain or tear domain. One thing to be careful of when looking at avian toll-like receptors is that there are regions of TLR1 and 2 families which are really quite clearly gene converted and very, very similar. And these regions should be excluded from various analyses. So if we see variation, do we see selection? And the answer obviously is yes. But we see that different TLRs express very different levels of positive selection and different patterns of residues are selected. So TLRs like TLR4 and 5, typically we see high levels of positive selection across a variety of residues in various different positions in those genes. Whereas with the nucleic acid recognizers, we tend to see that they're less variable, but they're less variable than those that recognize other patterns. And when you see variation, it tends to be focused on just one or two particular amino acids. Rather than a feature of the sort of strength of selection in these situations, I think in many ways this is probably a feature of the variation in the agonist or ligand, where non-nucleic acid-based agonists actually can vary quite widely compared with nucleic acid-based agonists, where there's more restricted structure and probably more restricted residues that can change to alter recognition. If we take those selective polymorphisms and we drop them and we map them against a phylogeny of penguins, what we can see is that different toll-like receptors, the polymorphisms in different toll-like receptors can be tracked back to particular groups of penguins. For example, with TLR5, there are different types of sequences that are more associated with different groups of penguins in their phylogeny. I now want to move across, rather than the sort of deep stuff of dealing with lots of different penguin species, I want to move to within species selections patterns, and in this case, the gentoo penguins. So why on earth did we pick the gentoos? We picked them because they have a circumpolar distribution. They're very widely distributed. They're found in a wide range of different environments, both north and south of the polar front, so in really quite different conditions. And this was essentially a sort of first attempt at looking at penguin TLR biology. So if we started off, we looked at TLRs 4, 5, and 7, the whole sequence of the TLR, not just a fragment, the whole sequence of each of these TLRs. And we found, just as we found with pan-penguin patterns, that TLRs 4 and 5 were had a lot more variation than TLR 7, and there were a lot more residues that were being that had evidence of some sort of selection. So this is mapping some of those residues onto the TLR, onto schematics of the TLR structure. And the first thing that comes out, just as I've said before, is most of those residues map on the extracellular domain, on the loose-enriched repeat domain that interacts with pathogen components. But with some of these TLRs, we got some really strongly selective polymorphisms that map to the signaling domain, in particular with TLR5. And that really led us to investigate this a bit more. So with TLR5, the two signature selective polymorphisms I'm going to look at are the 667 polymorphism and the 845 polymorphism. One's in the transmembrane domain and one's in the tear domain. Now, one of the reasons we selected them is these are two of the most highly selected polymorphisms. And actually, we had haplotypes of TLR that had either had these pairs of amino acid residues in very different configurations. So if we look at the Crozet gentoos, which are considered the ancestral, to represent the ancestral population of gentoos, then these had one particular haplotype, and these gentoos then migrated in a sort of clockwise spiral to end up with a southern clade of gentoos, which are subpolar front. And in these gentoos, we saw a dominance of a different haplotype of TLR5 that was essentially a derived form, and almost almost one haplotype made up all of the sequences of TLR5 we obtained. Now, we were very interested because this haplotype and this haplotype only had the two changes in that I've just noted. So the only difference between this haplotype and this haplotype are the signaling, are in the transmembrane and the signaling domain. So we couldn't resist it. We cloned both, and we asked the simple question: do they respond similarly or differently? And actually, what we'd expected is that this derived form might respond less strongly than the ancestral form. We were completely wrong. The derived form is actually a stronger signal than the ancestral form. So what might be happening? And there's lots of possible explanations here, but actually there's clearly selection as these penguins that are in the southern clade for this derived form that signals more strongly. Now, maybe it's that there's a particular pathogen hanging out here, or a pathogen that has greater effect hanging out here. But it may also be that actually the penguins in this location are actually not being challenged by a pathogen, that if you've got overactivation, you'd end up with disease. So we don't actually know the reasons. We don't know enough about the pathogens in the gentoos in these locations. We're trying to determine that at the moment. Right, so let's just move on to the sort of last bit of the talk about pathogens as selective forces. It's pretty complicated, to be honest. You've got to think of pathogen profiles, not single pathogens. Occasionally it's a single pathogen, but often it's a group of pathogens. And think agonist, not pathogen. More than one pathogen can combine to produce an agonist that is recognized by a single TLR, for example. Mixed use of pathogens can complicate the signals of selection, particularly if their agonists are binding different parts of maybe even the same TLR. Obviously, virulence is key, but remember, this might be immune-mediated virulence. We've already talked about how dangerous toll-like receptors are. Overactivity can clearly be as deadly as a lack of TLR recognition. Consider the mechanisms that TLRs are existing in, the mechanism of immunity as a whole, not just the target PRRs, but other parts of the immune system. The system acts as a whole, so this is quite complicated to think about, but actually TLRs don't function as the sole effective mechanism against any infections. So let's think about the pathogen perspective here. So if we take a single pathogen, in this case, Salmonella enterococci typhimurium, multiple TLRs recognize this pathogen, including TLR2, 4, 5, and 9. And they recognize different Pamps from Salmonella. So what happens if you take away these different TLRs? And this has been done obviously in mice with knockouts. And actually what's quite interesting, as I mentioned before, if you knock out TLR4, the animals get sick and die. But they're resistant to injecting the LPS, which is what TLR4 recognizes. If you knock out TLR9, you get increased infection, you get reduced survival, but the phenotype's nowhere near as bad as with TLR4. If you take out TLR5, which recognizes flagellin, you get reduced inflammation, you get reduced T-cell responses, particularly against flagellin, but you get no enhanced death. You get a completely different consequence. And then if you knock out TLR2, you actually end up with increased survival. And that's because you end up with more natural killer cells that produce more interferon gamma that drive more nitric oxide that kills the infection. So in this case, TLR2 is actually is detrimental to the host in some ways rather than driving host immunity. So it's not just that there's multiple TLRs involved in recognition, it's actually the consequences of TLR activation can be both positive or negative. The other thing to think about is that Pamps, they might be conserved, but they do vary. They're conserved, but not identical. And this can be really important. So lipopolysaccharide structure varies, and that affects TLR4 MD2 recognition. Flagellin structure varies, and that affects TLR5 recognition. For example, Salmonella and Campylobacter flagellins are quite different. And also the optimal unmethylated CpG recognition patterns of things like TLR9 differ very, very, very widely across different species. And the same looks to be true for avian TLR21s too. And I'm just going to sort of finish off with saying, well, actually, this interaction with pathogens is not a simple, stable environment because we do see changes in pathogens and changes in virulence can affect selection pressures. So one example here is the myxoma virus, causes myxomatosis, really very, very famous. I'm not going to go through this in detail, but really at the end of the day, it's not just the host became more resistant, and that's a polygenetic profile, but it's also that the virus became less virulent, and those less virulent strains gradually became more prevalent because they had more opportunities to transmit. And actually, we know what happened in these less virulent strains. Actually, it was a reduction in the level of antagonism of one of the intracellular pattern recognition receptors. So not a TLR in this case, but a pattern recognition receptor. So inhibiting this pattern recognition receptor was less strong in the less virulent strains. And we couldn't finish a comparative biology sort of seminar without considering COV2 under these current circumstances. And of course, comparatively, we have to think about coronaviruses in bats. And I'm not going to go through this slide in huge detail, but actually the real phenotype that you see across a range of bats is that they inhibit viruses getting a foothold in the first place, typically by using interferon-based, type 1 interferon-based pathways. But at the same time, they reduce inflammation. And this is where the pattern recognition receptors seem to be involved. There's a less active inflammasome, less activity in LLRP3. They don't seem to have the PIHIN locus, which includes one of the pattern recognition receptors, AIM2, and they have less active sting. And that reduces pathology, but it also reduces adaptive immune responses as well. So bats seem to sort of have a reduction in the early phases of viral infections. They tolerate low levels of infection by not getting disease, immune pathology, but they have a reduced ability to clear these infections. So that's me taking you through a whistle-stop tour of TLRs and other pattern recognition receptors. So in summary, what we think is these receptors are really good targets to understand immune selection, but they're not the only ones. Their patterns of selection can be complex, particularly in wild populations, and we have to think about pathogens and the host and how they might have different effects. We have to think about changing pathogen profiles and changing selection pressures. So finding the evidence of selection in some of these pattern recognition receptors is not so difficult, but the functional studies are what's needed to enable accurate interpretation. I hope I've convinced you of that. So I'd like to sort of end with a situation of functional immunogenetics. Yes, we need more sequence data. Yes, we need more sequence data within populations, not just a single example per species, but they need to be integrated with functional assays for us to be able to understand the way that these receptors are adapting to the environments. So thank you very much. And there are a number of key players, both people from within my group and many collaborators across the years. So thank you for listening, and I hope I've been somewhere near time.
Thank you, Ed.